Good morning, everyone. So I'm going to start off with the discussion question from last time. If you've been doing all of these, you'll see that the numbers are packing up for the final exam. So these are all worth a point on the final exam. So the question is, in patients who have deletions in AID, right, activation-induced deaminase, you can get this in autosomal recessive mutations, meaning you have to have two copies of the bad version of the gene. And those patients are susceptible to bacterial infections. And when you look at their lymph nodes, they have massive germinal centers. They're extremely large germinal centers. And the question is why? What do you expect the B cell phenotype to be? And what do you expect the T cell phenotype to be? And so when you look at this, remember what AID does. It's an enzyme that just takes Cs and converts them to Us. And then your B cells will try and repair that. And so it controls both somatic hypermutation, where it's making just a few changes, and it also controls isotype switching, right? Where the S transcripts are holding open the R loops, and AID gets lots of access there, so it makes lots of C to U transitions. So without AID, you don't have somatic hypermutation. And if there's no somatic hypermutation, there can't be a selection process because nothing is better than anything else. And so there's no affinity maturation. And that's why they have these massive germinal centers, because there's no competition, so everything just keeps going along. Okay, so for B cells, you're going to have low affinity antibodies. Now, the second thing is the AID is also controlling isotype switching. And so if you don't have AID, you don't get isotype switching, so you have hyper IgM syndrome. Okay, so for B cells, it matters a lot. The question is, does it matter for T cells? And the answer is no, not really. The T cells will go ahead and do their job, but without antibodies, it's very difficult to control infections. Okay, so last time we covered CD8 T cells, which are simpler. They get activated, they proliferate, they differentiate into effectors or CTL, and then they leave the lymph node to go and hunt out target cells. And so they're actually kind of simple. They basically have one job, which is to kill infected cells or tumor cells, and that's it, okay? People have tried to ascribe other roles for regulatory CD8 T cells, but that's not their program. They're really programmed just to go out and become effector killer cells. But when we talk about CD4 cells, it's much different because CD4 cells, remember, are the helpers, or a more appropriate term would be the controllers. So they control the immune response. And this was first observed in the 80s, in the 1980s, in mouse experiments where you just infected different strains of mice with Leishmania. And so if you infect them with Leishmania, if you infect Balb C mice with Leishmania, they're susceptible to that and they die. But if you infect other strains of mice like C57 black 6 mice, they'll survive. And the question was, why is that? And so the experiment was done to take Balb C mice and pre-treat them with an anti-interleukin 4 antibody, or anti-IL-4. So this blocks a cytokine that we know is important for some helper responses. And when you do that, the mice become resistant to infection and survive, which says that IL-4 is a key controller or a key signal for resistance or susceptibility to this bacterial infection. Now we know through whole genome sequencing, as well as other studies, that people have mutations in genes, okay? And it's not always just completely knocking the gene out, but it alters how stable it is or how much is expressed. And so in an outbred population like humans or wild mice, you have individuals that differ in their susceptibility to infection, to allergy, to autoimmune diseases. And that is in part regulated by differences in the ability of the immune system to generate different cytokines. And one of those is IL-4. And so here are some differences that have been noted. Okay, so if you have increased levels of IL-4 or increased IL-4 signaling, you become susceptible to leishmania, to Mycobacterium tuberculosis, as well as other diseases. Okay, so having too much of those makes you susceptible. On the other hand, if you have too much of other cytokines, like, too little of other cytokines like IL-12 or interferon gamma, you also become susceptible to those kinds of infections. Now, what does that mean? That's basically saying that there's two opposing things. More IL-4 makes you susceptible, but less interferon gamma also makes you susceptible, suggesting those are competing. There are also some others. More recently, it's been shown for IL-17 that if you have too much of that, you can get susceptibility to infection, or if you have increased levels of suppressive cytokines like tumor growth factor, I should say beta, not gamma. Sorry, this should be a beta. And so the idea is that you have different types of helper responses that are characterized by these different cytokine pathways. And if you make the wrong one, then you become susceptible to certain diseases. This was really shown sort of experimentally in a natural experiment, I suppose we'll call it, in 1969. So this was at the height of our vaccine making arrogance, where we thought, well, we can just make any vaccine to anything by just growing up the wild type virus and dumping it into formalin to kill it. And so they did this in 1969, and they used it to immunize a lot of preschool age children. So respiratory syncytial virus normally doesn't cause serious disease. It causes runny nose and sort of congestion. So there's an economic cost, right? Mom or dad has to stay home and take care of the kid. They lose out on work. And so it's a vaccine for something that's not a very serious disease. However, what happened is they gave the vaccine to the kids and everything was fine that year. But the next year, when those kids got infected by the virus, they had much more serious disease, and some of them were hospitalized and a few died. Okay, so this is a case where you actually bias the response to the wrong kind of response, and that actually impeded the children's ability to control the virus. Now, this has been proven in ferret models where you can vaccinate ferrets with the RSV vaccine and block this enhancement of disease by giving an anti-IL-4 antibody. Okay, so what's happening here? Well, formalin-fixed vaccines are weak, and they primarily give a Th2 cytokine response. And we'll go through what Th2 means, but that's sort of an IL-4 dominant cytokine response. And so the vaccine is skewing the T cell response away from a protective Th1 cytokine response, which is IL-12 and interferon gamma. And so the vaccine actually is sort of giving a handicap to the immune response to the wild type infection. Okay, so let's go into how do CD4 cells differentiate. And again, if we look at the differences between B cells and T cells, B cells differentiate at the level of the BCR or the antibody, right? They're changing the sequence, they're changing the isotype, but essentially B cells don't differentiate at the cellular level into lots of different kinds of cells. T cells are just the opposite. T cells don't change the T cell receptor at all. There's no somatic hypermutation. There's no isotypes. It's not secreting it. It's just the same T cell receptor as on the naive cell. What they change is their transcriptional machinery to become the right kind of T cell. So you get lots of different flavors of CD4 T cells. So we go back to the idea of how do you activate a T cell? Okay, so the main way, remember, is that dendritic cells, which are the cells that are mostly responsible for initiating T cell responses, are going to pick up antigen in the periphery, if they're migratory dendritic cells, and then go to the lymph node and activate T cells. Or if the antigen's getting to the lymph node independently because it's small and diffuses through the lymphatics, through the follicular reticular network, then lymph node resident dendritic cells pick up that antigen and present it to T cells. Now, I showed this slide before. There's lots of different types of dendritic cells, and some are better at activating CD4 cells, or some others are better at activating CD8 T cells. You don't need to know that for this class. Just know that all the dendritic cells can activate both naive CD4 and CD8 T cells, and that they're doing it in the same way. It's antigen, co-stimulation, and cytokines. But the cytokines here for CD4 T cells become important because it decides what kind of CD4 T cell you're going to get. And so here's the four main groups, and there are others. Okay, so the first ones that we knew about were the Th1 and Th2. So we just called them 1 and 2. And then Ethan Shivak and others discovered regulatory cells, so we called those Tregs. And that's fine. But then in the early 2000s, Laurie Harrington, working at the University of Alabama, Birmingham, discovered another subset of T cells that make a completely different set of cytokines. And we call these Th17. Why? Because they make an interleukin 17. Should they have called them Th3? Probably, but we didn't. So that's where we get the names. So those are the four main ones. There are also Th6 and Th9 and some other ones, but they're really sort of, it's not clear what their biological role is in vivo. So we have a naive T cell. It gets activated by a dendritic cell. It gets the antigen signal. It gets a co-stimulatory signal, and then it gets a cytokine signal. And one of those cytokines is IL-2 that allows it to proliferate. But then the other cytokines that are signaling that cell tell it what kind of T cell to become. Okay, so the signals from the dendritic cells, as well as other innate cells that are in that environment, are informing that T cell, this is the type of infection that we think we have, so you need to become this kind of helper cell. And so they have different outcomes. Th1 cells will make things like interferon gamma, which is good for activating macrophages and K cells and CD8 T cells. It's really directing it to say it's probably a virus or an intracellular infection. So you need lots of CD8 cells, and then you need to activate all of the other parts of an antiviral response. A Th2 response is really saying, well, this is probably an extracellular pathogen. And so you need to make antibodies against that. And that includes activating your granulocytes and alternatively activating macrophages for a different role. And that's really saying, we think this is going to be extracellular, so we need these granulocytes to be ready to degranulate towards that extracellular pathogen. Th17 cells, on the other hand, are primarily in mucosal lymphoid tissue, and their job is to activate cells to make a mucosal response. And so you can start thinking about the type of antibodies it would direct, but it's primarily IgA that's going to push things back at the mucosal surface. And then finally, Tregs are inhibitory cells, and they will inhibit other T cells, and we'll talk about how they do that, but they'll also shut down dendritic cells. There was just recently shown by a group at NIH that they can actually steal MHC off of those dendritic cells so they don't have anything to present to other T cells and shut down the initiation of other responses. Okay, so we're going to stick with these four main helper cell subtypes. We won't discuss the other ones that are possible, but they're really just different flavors of these. Okay, now one important part of this is that now you start activating these other cells and there's a feedback. And so, excuse me, as you activate eosinophils and basophils, your granulocytes, they start making more of the cytokine that directs a Th2 response. Whereas if you have activated macrophages, NK cells, and CD8 cells, they also make interferon gamma. And so that directs this response towards this side. Okay, so if you have these cells, you're going to reinforce this pathway. If you have these cells, they're going to reinforce a Th1 pathway. Okay, so it's amplifying the type of response that you get. So how does this start? Okay, so initially we have our dendritic cells sitting out here in the periphery. Okay, this is, you get an infection in your big toe, that dendritic cell is going to start, it's going to take up antigen and get PAMP signals, and that's going to tell it to go to the lymph node and start activating T cells. Well, if you remember, you really can break down PAMP receptors by where is the pathogen. Okay, so if it's a virus, here's our virus, oops. Here's our virus, it's getting in. If that dendritic cell is going to take up that, it's going to get PAMP receptor signals from things like, you know, the endosomal TLRs, the Rig-like receptors. That's telling the cell this is an intracellular pathogen, and you need to make an appropriate response for intracellular pathogens. In that context, you also have other cells that are sitting there that are going to make cytokines, okay, to influence this. So NK cells, excuse me, and NKT cells will make things like interferon gamma. That is directing the response to become a response against intracellular pathogens. Excuse me. However, if it's something like a parasitic infection, here's our worm getting in, that's going to stimulate a different set of innate cells, in particular your granulocytes, and that they're going to make cytokines that tell this cell, yeah, you should become a different kind of, or you should direct the response to be against extracellular pathogens. And if this dendritic cell is in that environment, it's going to get PAMP receptors from extracellular toll-like receptors. Okay, so it's sensing either lipopolysaccharides or different forms of lipids or sugars that it can recognize on that parasite. Okay, so now those initial signals, both the PAMP receptors and the environment, are giving instructions to this dendritic cell saying, when you go to a lymph node and start presenting antigen to your T cells, you need to direct the response towards that kind of infection. And so here we have, this would be called a Th1 response, okay, where it's characterized by cytokines that tell T cells you should become a T cell that directs, that further directs us to an intracellular pathogen response. On this side, we have a Th2 response, okay? And it's really saying the dendritic cell is getting these instructions, and so when it presents to a T cell, it's actually making cytokines that tell the T cell, you need to be directing the response towards extracellular pathogens. Okay, so that's the easiest breakdown. If it's intracellular, you have this one set of toll-like receptors and Rig-like receptors and cytokines that instruct the dendritic cell that then can instruct the T cell. Okay, so if we go back to cytokines, it's important to think about how these work, okay? And so cytokines, most of the cytokines that we'll talk about today work through the JAK-STAT pathways, okay? So the cytokine will bind to the cytokine receptor on the surface of either the dendritic cell or the T cell. That recruits the Janus kinases. Remember, this is the Roman two-faced god, and they phosphorylate each other. They then phosphorylate the receptor, which recruits the STAT proteins. Those get phosphorylated, and what they do is then they dimerize. The phosphorylated STAT proteins dimerize, and that allows them to go into the nucleus and act as a transcription factor. Okay, so the cytokines are turning on genes. These are very simple pathways. They're basically, each cytokine receptor uses a particular JAK, a set of JAK proteins, and a set of STAT proteins. And these are specific for each cytokine receptor. So when we look at this, first of all, there's the first cytokine that we talked about was interleukin-2. This is the T cell growth factor, right? And that's made in all cases. You always get IL-2. Once a dendritic cell stimulates a T cell, it's going to start making IL-2. But IL-2 will affect a number of other things. First of all, it will upregulate Th1 and Th2 cytokine receptors. In this case, the main ones that start a Th1 response are IL-12 binding to the IL-12 receptor. So here IL-2 is turning on STAT5. That then will cause the transcription of the IL-12 receptor. Then sort of takes it so it can respond. But it's also going to upregulate the IL-4 receptor. And so this is sort of in a computer programming sense, it's sort of like saying, okay, I got a signal to proliferate. What should I do now? Okay, so it's a query. What cytokines are present there? Now, IL-2 will actually suppress the cytokine receptor for Th17 responses. We'll talk about why that is, because really Th17 responses are designed to be plastic so that you don't always, another way of saying that is, you're always making Th17 responses, but most of the time it's not inflammatory. They're just sort of making antibodies to push back stuff in the microbiome environment. And so you don't always want those to be proliferating into this massive response. On the other hand, IL-2 does not affect Treg cytokines. And part of that is because IL-2 is the Treg cytokine. Okay, so the IL-2 receptor is part of regulatory cell pathways. Okay, so we'll talk about that in a bit. But essentially what I want you to see is that once a T cell gets activated, it starts expressing these other receptors, okay, these other cytokine receptors. And depending on what cytokine signal that cell gets, if it gets IL-12, it'll become a Th1 cell. If it gets mostly IL-4, it'll become mostly a Th2 cell. If it gets IL-6, which is another cytokine we've talked about, it'll become a Th17 cell. And then regulatory cells are sort of counteracting this by trying and binding all of the IL-2. They're trying to act as a sponge. But signaling through any of those cytokine receptors is going to activate specific JAK proteins that lead to the activation of specific STAT proteins. Now for this class, you don't have to know the Janus kinases, okay? I'm just asking you to know the STAT proteins because those are transcription factors. And what they do is they turn on other transcription factors, sort of master control transcription factors. And this commits that cell to becoming that type of T cell. So Th1 cells are programmed because STAT4 turns on a transcription factor called TBET. Th2 cells become programmed to become a Th2 cell because STAT6 will turn on a transcription factor called GATA3. Th17 cells become committed to becoming a Th17 cell because STAT3 will turn on a transcription factor called ROR gamma T. And finally, Treg cells, induced Treg cells, are going to turn on transcription factor called FOXP3. Okay. Now look downstream of that. These are all programmed now. These are master transcriptional regulators. And so they start producing cytokines that reinforce this pathway. The Th1 cell, if it gets a signal for IL-12, will start making interferon gamma. That feeds back, binding to the interferon gamma receptor and reinforcing that pathway. Th2 cell will start making IL-4. That will bind to the IL-4 receptor and reinforce the Th2 pathway. Okay, and there are a number of others, and we'll go through these each of the cell types independently or separately throughout this lecture. Now, what we do know is that the act of those T cells being programmed to become a particular helper type, they start making these cytokines. Those actually block the other side, the other helper responses. Okay, so a dendritic cell here is activating a naive helper T cell. Okay, and the main signal it's getting is IL-12. That tells that cell, okay, signal and turn on TBET, which is this master transcriptional regulator. TBET then turns on things like interferon gamma. Interferon gamma blocks or turns off transcriptional programming for Th2 type cells. Okay, so it's saying, well, we think that this is a Th1 response, so we're going to do this, and we're going to try and shut off everybody else. And you think of it as a sports game, right, where you're trying to defeat the other team because you think that you're right. On the other hand, if you have a dendritic cell and it's providing IL-4 initially, that tells this naive T cell, when you become activated, you're going to turn on GATA3, and that then will control production of IL-4. And IL-4 will block the Th1 program, okay? So it blocks cells from going down this pathway. So these initial signals are really saying, which way should we go? And it really becomes, which one becomes more powerful? If there's lots of signals become a Th1, that eventually shuts down any Th2 cells from working. If it's a Th2 response, those will shut down the Th1 cells from keep going.
Dr. Blattman?
Yes.
What if they're all present at the same time? Is there like a certain level that they need to be at in order to block Th1 or Th2?
The truth is they're all present at the same time every time.
Okay.
When you first start the response, you get Th1, 2, 17, and Tregs. And it's only in that environment they're competing. And they're saying, well, this is the dominant signal, it starts shutting down everybody else. Okay, so it's really what is the strongest signal in that group. And this is what it sort of looks like, right? Is if you include all of the transcription factors. Here's our naive T cell. And if it gets IL-12, binds to the IL-12 receptor, that signals through STAT4, that will eventually turn on TBET. Okay, but that's also reinforced that T cell then, TBET is turning on interferon gamma, which then binds to the interferon gamma receptor, signaling through a different STAT protein to reinforce TBET expression. Okay, so it's sort of, for Th1 cells, it's sort of IL-12 initially, but then interferon gamma reinforces it. For Th2 cells, they get an initial IL-4 signal that binds to the IL-4 receptor, signaling through STAT6 to turn on the master transcription factor, GATA3. GATA3 turns on IL-4. And so it's IL-4 initially and IL-4 reinforcing it. Now, for Th17 cells, this also works, but they're not directly competing, right? IL-17 or Th17 cells are a little more plastic in what we want them to do, and we'll talk about them in a second. But some of the signals that they're getting, or the main signal, is IL-6. IL-6 will bind to the IL-6 receptor, signal through STAT3 to turn on ROR gamma T, and then it starts making cytokines like IL-23. Okay, IL-23 will reinforce this pathway, reinforces ROR gamma T. Okay, so you really have these interferon gamma from Th1 cells will shut off both Th2 and Th17. IL-4 from Th2 cells will shut off Th1 and Th17. But Th17 cells are sort of the ones they don't care. They're not really competitive, so they're going to do their own thing. They don't really try and inhibit anybody else. Now, what we've talked about, this lineage commitment, where it's you turn on that transcription factor, either TBET, GATA3, or ROR gamma T, that programs a cell, that commits that cell to becoming that type of helper cell. And this means a Th1 cell cannot become a Th2 or a Th17. That's the only thing it can do. Well, that was originally defined in mice, and that's true for mice. But now when we look at human cells, they're a little more plastic. Like if you start off as a Th2 cell and you get enough interferon gamma signal, you can actually come over here and become a Th1 cell. So you can have a conversion process. That's plasticity. So a Th1 cell can become a Th2 or Th17 if they get enough other signals. Okay, I don't care that you really differentiate between those two, because in reality, in order to become a different type of helper cell, you have to provide extraordinarily high cytokine signals, and it's not clear you can ever really do that in vivo. So even if they are a little plastic in what they can become, it's probably not happening during an actual infection. Okay, so we talked about all the other costimulatory proteins, right? CD28 is the main costimulatory protein that will turn on CD40 ligand. That sort of biases the cells to have lots of proliferation. If there's very strong signals, biases it towards a Th1 response. On the other hand, the CTLA4, right, which is opposing CD28, this should have an X through it, is going to block Th1 responses and instead prefer to make regulatory cells. Okay, so if you can't get a CD28 signal because CTLA4 is blocking it, you instead get a regulatory cell. But now there's all of these other costimulatory proteins. Okay, and they're basically, their job is to say, help that T cell decide what kind of T cell it should become. Okay, so if you get a CD30 signal, that says, yes, the dendritic cell is pretty sure this is a intracellular infection, so you should make a Th1 response, okay? Th1, another way to think of it, Th1 responses are really for, this is a real full-blown 4-alarm fire infection, let's just go. On the other hand, if you get OX40 signals or ICOS signals, these are really biasing the response towards a Th2 response. And they're also telling the cell, you need to make some follicular helper cells. And we'll talk about that in just a second. So you can see that they sort of go together. Here you have follicular helper cells, and here you have Th2 cells. Okay, so these other co-stimulatory signals are really directing the CD4 cell to tell it what kind of response do you need. Then finally, there's a co-stimulatory protein called 41BB that's directing the cells to become Th17 cells. Okay, so this is in addition to the PAMP receptors on the dendritic cell, cytokines from the dendritic cell. The co-stimulatory proteins are also telling the T cell what to do. Okay, now, though another way to think of this is the PAMP receptors are telling that dendritic cell you should make these cytokines and these co-stimulatory proteins. That will help bias the response. Okay, so it does influence it. It's less absolute than cytokines, but it certainly has a role in shaping the T cell response. So here's our sort of simplified view. We've added a fifth group here. We have Th1 cells. Those are characterized by the master transcription factor TBET. You start that off with IL-12, but it's reinforced with interferon gamma. Here we have our Th2 cells. Those are characterized by the master transcription factor GATA3. That's induced by IL-4, right? That then causes that cell to make IL-4 and reinforce that pathway. Now our Th17 cells are really influenced by IL-6. And so that acts on the Th17 cells to turn on the master transcription factor ROR gamma T. And then those cells will make lots of other cytokines, including more IL-6 and IL-23. Okay, that reinforces that pathway. Now, the fourth one that we talked about were the regulatory cells. And this is really if you get CTLA4 blocking co-stimulation, you're also making suppressive cytokines like TGF-beta. That tells those cells to turn on the master transcription factor, FOXP3. That then tells that cell to make more TGF-beta that reinforces that pathway. So it is doing, there in all four cases, you're sort of doing the same thing. But often then we include another subset, and these are the follicular helper cells. Now we've already discussed what their job is, right? They sit in the germinal center, in the light zone, and they're providing CD40 ligand signals to B cells. The B cells have CD40 on them. For those B cells to re-enter the dark zone, start redoing somatic hypermutation, isotype switching. So the follicular helper cells are initiated during a T cell response. And it could be from OX40 or other costimulatory proteins, but it's really this cytokine IL-21 that is directing these cells to become follicular helper cells. Now the thing is, if you get a Th1 response, you get Th1 biased T follicular helper cells. If you have a Th2 biased response, you get Th2 biased follicular helper cells. If it's Th17 biased, you get Th17 biased follicular helper cells. The question is, if you get a regulatory response or Treg response, do you also get regulatory follicular helper cells? The answer is no, because what they're actually doing is shutting off dendritic cells, shutting off the response, so you're not getting it. They're trying to suppress all of the T cells, okay? So we've included our fifth group here, but keep in mind, this fifth group is just, it has a different job. Its job is to go into the germinal center. And so it can be a Th1 bias, can be Th2 biased or Th17 biased, whatever kind of signals it's getting to become that kind of follicular helper cell. Okay, so what do these cells do? And we're gonna start breaking these down into the different groups, right? So Th1 cells, those are induced by an intracellular infection. Okay, so you get intracellular PAMPs that are directing that dendritic cell to make IL-12, and then it will go and cause T cells to become T-bet programmed, and their job is to go and suppress intracellular infections. One of the ways that they do that is they make interferon gamma, okay? By its name, interferons interfere with infection. And they can do this in a number of ways. For macrophages that have, in this case, this would be endosomes containing bacterial infections, that's still intracellular, and so it will cause the macrophage to fuse those endosomes with lysosomes to kill what's ever in them. So it's an activation step, okay? The interferons will also shut down cells to make them resistant to infection by viruses. So there's this global response. And it's another way to think of Th1 cells is they sort of are wannabe CD8 cells. They go out, they try and make these cytokines that suppress infection, and they actually can kill to some degree. Th2 cells, remember, are initiated from an extracellular infection. So something like a parasitic worm. And so those cells are going to leave the lymph node and make cytokines like IL-4 and IL-5. And these are going to activate the granulocytes. So they influence mast cells and eosinophils. They'll also, we'll come back to this, they're also influencing the kind of antibodies that you're making. It's going to direct, or actually it's influencing the amount of antibodies you're making. So it's going to direct the B cells to become more geared towards antibody production, because that's what you need. You need lots of antibodies to coat that in extracellular pathogen so your granulocytes can kill it. Th17 cells, again, are typically in mucosal associated lymphoid tissues, and they're going to make cytokines that are appropriate for telling that the cells in that area, well, you need to either change the structural architecture so it can work on epithelial cells or fibroblasts. and help them rearrange to sort of seal any breaks in the epithelia. It also activates neutrophils that come in and sort of, if there's an infection there, they'll respond to it by exploding and decorating stuff with their extracellular nets. Okay, so we'll come back to Th17 cells later in the lecture, but they're really in mucosal surfaces, and so you don't want them to be making huge T cell responses and killing everything. They're more important for maintaining structural integrity. Now, again, we have our follicular helper cells. They, whatever kind of response you get follicular helper cells that go to the follicle. They recognize antigen on B cells. They provide the CD40 ligand signal to CD40 on the B cell. That tells the B cell to do isotype switching, should say somatic hypermutation, but then that results in affinity maturation. Okay, so no matter what you get a follicular helper cell. And finally, our regulatory cells' job is to shut down T cell responses. And so they'll make things, cytokines like TGF beta. Look like a 4-year-old trying to write here. Those will suppress dendritic cells and shut off any kind of T cell response. Now, if it seems like I'm saying the same thing over and over, it's because in part I am. We're sort of hammering out the main highlights of each of these different kinds of responses. So when you look at one way to look at this is what's the strength of the infection, right? How much do you want to commit to this? And so there is a difference. The very strongest responses, or where you get the most T cell proliferation, the most IL-2, are the Th1 responses. And that's another way of saying that is viruses in particular replicate many orders of magnitude faster than you can, and they're actually replicating faster than bacteria because it's not just a division. They make lots and lots of themselves, millions of themselves from any infected cell. So to control a viral infection, you need lots of a Th1 response. Okay, so it's a full-blown proliferate as much as you can so we can try and clear a viral infection. A Th2 response is typically a response to extracellular things like parasitic worms, low levels of bacterial infection. Well, if you get a parasite infection, you're really talking about one or a few parasites in the entire body. So you don't need a huge response for that. What you want is just enough. So you don't have to commit to lots of proliferation. And so you have much less IL-2 production and proliferation for a Th2 response. Okay. Now, regulatory cells, well, you don't want a response, so you shut it down the most. And Th17 cells are sort of these mucosal responses, so you want them to be there, but you don't always want a massive inflammatory response. So it really limits their proliferation. Okay. So what do I want you to know from this? Again, we're just sort of adding on, how does the immune system know which kind of response to make? And it really boils down to, well, how much of a potential problem could there be? So now let's go through the different helper cell responses individually. And we'll start with the Th1. Right, Th1, as I said, they're sort of wannabe CD8 T cells. So here's our dendritic cell, comes into the lymph node. It's got an intracellular pathogen, or it's gotten intracellular PAMP receptor signals. It's gotten cytokines from that environment, from NK cells or NKT cells that tell it, yeah, you should make a Th1 response. And so it's going to be providing IL-12 to the CD4 cells. Now remember, that pathway is reinforced by interferon gamma. Well, at the same time, you're going to have dendritic cells that are going to activate your CD8 T cells. Okay, so those proliferate, they leave the lymph node, they go out, and they're looking for things that are infected. If they recognize something that's infected, here's a cell with a virus inside of it, they just kill it. Th1 CD4 T cells are essentially trying to do the same thing. CD4 cells will recognize the dendritic cell. They'll get IL-12 signals that tells them to be Th1 cell. They proliferate, then they leave the lymph node, and they're going and looking for infected cells. Now, they can kill that infected cell, so they can act just like a CD8 T cell, mostly by using fast ligand pathway or the TNF receptor pathway. They don't really have perfect enzymes, so they're trying to kill, but they're not great at it. They'll also cause activation of macrophages and really relying on other cells to do the killing part, but they're trying real hard. Okay, they're going out and they're trying to be a CTL, but they're just not very good at it. But they are good at inflaming everybody else. So how do they do this? Well, there's a number of different ways, or a number of different things that Th1 cells will do. They get activated and they go out into the periphery and they see an infected cell or a macrophage that has something inside of it. They will provide interferon gamma. Okay, and that's this key cytokine for Th1 cells. They also will give CD40 ligand signals. And so that activates the macrophage and it will enhance its ability to kill what's ever inside of it. It's also doing this to all kinds of cells, not just macrophages. It's really saying, do I send something here? Can I help the CD8 T cells out by telling that cell to kill what's ever inside of it? Or in some cases, just kill the cell directly. Now, the other things it'll do is it'll activate a lot of the other sort of innate cells. And it does this through the tumor necrosis factor, TNF pathway. So the two main cytokines that a Th1 cell makes, the main one is interferon gamma. The second one is TNF. And you've already seen this. TNF signals on cells to either activate them or kill them, right? And for most of the cells in your body, it's going to tell them, yeah, you should undergo apoptosis. So it's going to activate caspase 8, and then downstream caspase 3, and then start that whole death pathway. But in some of your other cells, like neutrophils, it'll signal through the other TNF receptor and cause activation, okay? Now, as I said, if you get a Th1 response, you get Th1 biased follicular helper cells. And so those are here, right? These are your follicular helper cells. F, H. There was an easier way to do this. So here's your follicular helper cell. It's going to go to the germinal center and start providing signals to the B cells. Well, it'll make interferon gamma. Interferon gamma will turn on the IgG gamma S transcripts and have isotype switching to IgGs. And so you get production of systemic antibodies that are basically saying, there's an infection here. We want to start to neutralize, opsonize, and coat anything that's in the system so that it can be destroyed. Okay, so when we look at a little, and we go a little bit closer, and this is a figure I think it's important to sort of say, how's it doing this? So here's many types of bacteria have evolved strategies to just survive in the endocytic compartments. Mycobacteria is the sort of the original version that people knew about, but there's lots of other bacteria and some viruses can do this, where they just get in, they hide out and they don't do anything. They're just sort of waiting for their turn. And so to get rid of those, you have to tell that macrophage, you've got something inside of you, you need to fuse lysosomes with those endosomes to kill it. So to flush it out of the system, okay? So the main thing is happening is the T cell comes along, it recognizes the antigen on the surface of that T cell, so it's antigen that's being presented on MHC, and then it's providing a cytokine, in this case, interferon gamma, okay, that is telling that macrophage, you need to kill what's ever inside of you. We'll also produce things that are going to tell everybody else, hey, there's an infection right here, so come and join us in this little party responding to the infection. And that is things like macrophage chemoattractant protein. You have to remember that. No, you're just making chemoattractants that recruit additional cells. Now, what this does is in many cases, what it's trying to do is cause sort of a compartmentalization of the infection. And this is most clearly seen in mycobacteria, some Neisseria species, some fungi will do this, where what you're trying to do is wall it off. And so what happens is that you recruit all these cells, and they're sort of walling off the infection to form a granuloma. And so people who have a Th1 bias during mycobacteria infection will form granulomas, basically little nodules of infection, but they're surrounded by everything else, so the infection can't get out of it. If people are Th2 biased, they actually develop a leprosy type disease where they can't wall it off, it spreads systemically, and you start getting rotting tissues. Okay, so this is really important for many of the intracellular bacterial infections, because if you don't have it, you can't form a granuloma. And so lack of that results in uncontrolled microbial infection, and then you get a lepromatous disease. Okay, so that's sort of what the Th1 cell itself is doing out in the periphery, but I want to bring this back to the follicular helper cell. Okay, if you get Th1 cells that are making interferon gamma, you're going to get follicular helper cells that are also making interferon gamma. Remember what this does. Okay, interferon gamma is going to bind to the interferon gamma receptor on B cells to activate STAT1, and now you get IgG gamma S transcripts, primarily IgG2A and IgG3. Okay, those are the main ones for just antibodies that are good at blocking infection, neutralizing, opsonizing systemic infections. Okay, so in the B cells, you've got this S transcript is holding open the R regions before these two isotypes, and that allows APE1 to come in and recognize all of the CDU mutations that have been cleaved out and start making cuts there. And so this is going to cause the B cell to make lots of these damages in the R loops for those two isotypes. And that then will, when you repair it, now you get isotype switching to those two types of antibodies. You get IgG2A and IgG3. Okay, we're going to come back to that for each of the different cytokines or different helper cell responses. Now, as we said, if you become a Th1 cell, you're trying to block a Th2 cell. Okay, so you do that by, from cell to cell, it is doing this by cytokines. But within the cell, you're also trying to prevent that Th1 cell from becoming a Th2 cell. So you want to hold on to that. It is already a Th1, so you don't want it to change its program to a Th2, even if it gets that other cytokine signal. And so here's our antigen presenting cell. It's presenting to a T cell. If it goes down the Th1 pathway, right, then it'll be reinforcing this by having interferon gamma signals to STAT1 to reinforce Tbet expression. Well, you could imagine, well, what if that cell is now getting an IL-4 signal? Well, that turns on STAT6, and that's going to try and turn on GATA3, but Tbet is actively suppressing GATA3. Okay, so in the Th1 pathway, the master transcription factor is also blocking other master transcription factors. Now, instead, if this was a T cell that went down the Th2 pathway, and it's now, making IL-4, signaling through STAT6 to upregulate more GATA3, it's reinforcing it with IL-4. GATA3 suppresses Tbet. So even if this cell were to get an interferon gamma signal, that cell's already committed, yes, I'm a Th2 cell, okay? So this is where we get lineage commitment. Now, if you provide enough of this in vivo or in vitro, primarily it's been shown in vitro, you can overcome this, but really it's the transcription factors that are mutually inhibiting. Okay, so we've covered Th1 cells. When you think about them, what should they be? They're CD8 wannabes. Their job is to activate macrophages. They are going to further activate other immune cells like NK cells and neutrophils. But they're also making cytokines that are inhibiting infection, and they're trying to kill, but they're not very good at it. Okay? Th2 cells are a little simpler, right? And you have to remember, why did we get a Th2 response? Well, it's an extracellular pathogen, like a parasite. And so what's good at helping you kill parasites? Well, antibodies that are going to bind to it, that then your granulocytes can use their FC receptors to recognize that antibody. So you can have IgG antibodies. In this case, the two main ones are IgG1 and 4. You're always going to get some IgG. Okay, that's just part of the program. And that's because those are systemic antibodies. In this case, those are the ones that are being used by granulocytes to recognize this big parasitic infection. But the main one that you get is IgE, right? And remember, IgE is going to bind to high affinity receptors on mast cells, so that they use this as a surrogate receptor. They're going to be just display antibody on the surface and go and try and recognize this helminth or worm infection. If they do that, they degranulate and kill it. Now, a Th2 response, we said that it's trying to block a Th1 response. It will actually also do this by blocking macrophages, and primarily making a cytokine called IL-10. Okay, so this is a macrophage that thinks it should be doing something, but Th2 cells will shut down the macrophage and say, save your energy. We don't want you to make a response right now. What I want you to know about Th2 cells, they make this IgE, and that's being used to kill big extracellular parasitic infections. That's the main thing that they do. I know there's lots of arrows here, but that's really the gist of it is make antibodies that are going to help your granulocytes kill that worm. That's it. Okay, so this pathway gets reinforced by a number of different things. And the main one is IL-4. And I think, for the purposes of this lecture, I think I'm just going to take this out. Okay, so I won't ask you any questions on this. But these reinforcement pathways are telling the cell to do a lot of other things, like where it should go, what kind of things it should do, and also to survive. But I don't think you need to know any of that, and I won't ask you about this slide. But one thing that they do that is important or one of the ways that they're blocking macrophages is sort of a different way of thinking about this. So the Th2 cell will make IL-10. On the macrophage, right, this is the T cell producing this, the macrophage will bind the IL-10 and signal through STAT3, right? This is a different transcription factor, but that upregulates a protein called SOX1. Excuse me. SOX stands for suppressor of cytokine signaling, okay? And that shouldn't surprise you what it does. It suppresses other cytokine receptors. And Tregs do this very well, but Th2 cells will do this particularly for macrophages. And what suppressor of cytokine signaling 1 does is it blocks the STAT proteins. Oh, that's a terrible box. So it'll actually dephosphorylate the STAT proteins. And so then you don't get macrophage activation, can actually suppress T cell activation. So this pathway is can shut down other cells. Okay, so that's what the Th2 cells do. They primarily are involved in telling antibodies, you should switch to IgE, and they're also shutting down sort of Th1 type responses. Now, if you get a Th2 biased response, you're going to get Th2 biased follicular helper cells. Those will go into the germinal center and produce IL-4. Okay, and that will bind to the IL-4 receptor on B cells to signal through STAT6 and induce the main one is Ig epsilon S transcripts. Okay, so the Ig epsilon S transcripts hold open the R loop ahead of Ig epsilon, and you get to your CDUs transition there, and then you cut those out, and then APE1 will come in and recognize those apurinic sites and cause double-stranded breaks. And so then when you repair that, you get isotype switching to IgE. Okay, so it's the same thing that's happening for if you were to have interferon gamma, it's just inducing a different S transcript. Okay, so that's Th1 and Th2. We sort of, if you were to go and do a literature search of all the different things they could do, it would never end. You would have thousands, tens of thousands of papers to read. But in reality, Th1 response is making interferon gamma to activate macrophages and cells that will kill intracellular things. Th2 cells will make IL-4, which is directing to be an extracellular response, to be antibodies that are good for killing helminths and worms. Th17 cells are a little different, okay? And part of the reason is if you had, let's say you had eggs for breakfast. Eggs are a protein, source of protein. You're making a Th17 response right now to those eggs, but hopefully you don't feel any inflammation and you're not experiencing any discomfort because Th17 cells are not designed to make an inflammatory response. They have to get another signal to do that. And so instead what a Th17 cell is main job is to direct the production of mucosal antibody. The main one is IgA. So here we have our naive cell. It gets activated in the presence of IL-6. Should have the IL-6 here. That's the main signal to tell it to become a Th17 cell. And its job is to make cytokines that tell those B cells to make IgA. That's its main job. Now, your immune system doesn't always play by the rules, okay? If that's what's happening and there's no inflammatory signal, it will become a sort of regulatory Th17 cell. This is sort of in between a regulatory cell and a Th17 cell. It's basically saying, okay, we made a response, but there's no inflammation here, there's nothing to see, so let's shut it down. And so it starts saying, okay, there's nothing to do, and so it'll start making these suppressive cytokines like IL-10. IL-10 is going to turn on the SOX1 pathway and suppress other STAT proteins. Now let's say you've got this Th17 cell, it's directing an IgA response, but instead of getting those suppressive cytokines, it gets an inflammatory signal in the form of IL-12. Well, then it becomes more like a Th1 cell, okay? And it becomes inflammatory. And the thinking behind this, if we anthropomorphize this Th17 cell is, yeah, it's making IgA, it's telling the B cells to make IgA. But on the other hand, there's something here. Like I'm getting signals that there's an inflammatory reaction going on. So I'm going to try and help kill it. And so it starts making the Th1 type cytokines. And one of the main ones is interferon gamma. So really the Th17 cells, when they initially get activated, they're just telling the response to make IgA. And depending on whether they get inflammatory signals or not, they'll either become regulatory-like or they'll become Th1-like. Okay, and this is part of the reason that it took us so long to discover them is how do you get those without actually perturbing the system to one or the other outcomes. Okay, so when you're thinking about Th17 cells, they're really in your mucosal associated lymphoid tissues, primarily gut-associated lymphoid tissue, bronchus-associated lymphoid tissue, and their job really is to sit there and direct any B cells to make IgA. But if they get inflammatory signals, right, such as IL-12 or interferon gamma, Th1 type signals, they will start telling other cells, okay, there's something here, something's getting through the epithelia. We should definitely make a response. So they're going to activate macrophages and other cells to kill anything that's in that environment. Do you need to know all the way they do that? No. I'm just sort of keeping it high level what their job is. Okay, so inflammatory T cells, inflammatory Th17 cells are really only observed if you have microbes that have crossed the mucosal epithelia. And now that Th17 cell got an additional signal, yes, there's something here, we need to make a response to it. And this happens this also can happen during tumors or when you have a tumor. So here's our dendritic cell in a mucosal associated lymphoid tissue. It's activating CD8 cells. This can go and kill tumors. But you can also get in the mucosal associated lymphoid tissue, you can get Th17 cells. Okay. Now, if they're not inflammatory, their main job is just to regulate the structural integrity of the epithelial surfaces. This is shown sort of differently. It's maintaining the endothelial cell surface of a blood vessel. But then it will, if it's getting an inflammatory signal, then it is going to activate NK cells. It's going to help with CD8 T cells, and those are then going to respond to the tumor. Okay, so really it's Th17 cells are initially sort of neutral, and then they can be negative if they don't get any inflammation signals, or positive or aggressive if they do get an inflammatory signal. This is part of the reason why we think Th17 cells are probably involved in many autoimmune reactions, because you started off, you can just make a response to virtually anything, and it's neutral. But then you get some other infection or something else comes along to provide an inflammatory signal, and now that's going to override those cells to become more aggressive. And we'll talk about that in the next section of the course when we cover autoimmune diseases. Okay, so now we go back to this idea. Helper cells are controlling the antibody response. So if you get a Th17 biased T cell response, you're going to get a Th17 biased T follicular helper cell response. And so those follicular helper cells will make Th17 cytokines, in this case IL-21, and that signals through the same pathways. The main result is getting Ig alpha S transcripts that will hold up in the Ig alpha R loop. You get the mutations, CD mutations, they get cut out, and then APE1 comes in and causes double-stranded breaks. And then that causes isotype switching to IgA. Okay, so this is how the Th17 cells are directing B cells to become the right kind of antibody secreting cells. The IgA is then made, transported by the poly-Ig receptor across mucosal epithelia so that it can bind to things inside the lumen of the gut or the lung and prevent them from getting access. Okay, so that's Th1, Th2, and Th17. We're going to spend a lot less time on the two other subsets just because we've sort of covered them as we've gone along. But it's important for Tregs, right, that they got TGF-beta signals that told them to express FOXP3, and then they're making suppressive cytokines like TGF-beta and IL-10. So those inhibitory cytokines will shut down T cells, effector T cells. So that's the first way that Tregs suppress other responses, just by cytokine production. But they can actually do a couple of other things. What's often not really appreciated is Tregs have perforin granzyme. And so what they can actually do is go along and kill effector T cells. Where does this happen? It's mostly natural Tregs that are coming out of the thymus. Remember, if you're a developing T cell and you have super high affinity for a self-antigen, well, then you become a natural Treg and you go out and you look for other cells that have that and you're trying to shut down responses to those. And so Tregs can kill effector cells and sort of shut down the response that way. But those are sort of not as efficient as the other two mechanisms. The first is that they act like a sponge, right? An effector, a Th1 or 2 or 17 response is proliferating, so it needs a lot of things. It needs nucleotides, it needs proteins, it needs growth factor. And so the T cells will come along, and one of the earliest known functions was just they always express the high affinity IL-2 receptor. And so you're going to sop up all the IL-2, so there's none left for the effector T cells. Those cells don't have IL-2, then they die off. Okay, so this is one of the most efficient ways. You just take all the resources for yourself and you leave none for the other kinds of T helper cells. It can also do this in other ways. It can take up all the adenosine, it can take up the limiting amino acids, and basically say there's nothing for you to use here, and so those cells will die off, okay? And they'll go to here. So it's either cytokine starvation or starvation of other key metabolites. But the most efficient way that the dendritic cells can prevent other cells from working is to just shut off a dendritic cell. If you shut off the dendritic cells, they can no longer stimulate other cells. So there's a number of ways that they can do this. But basically, our Treg will come in, recognize antigen on dendritic cell, and it will start to shut that off by these other what are called inhibitory receptors. It will also make things like a very suppressive second messenger called indoleamine oxygenase, which will then it will catalyze tryptophan. If you don't have enough tryptophan, you can't make proteins, and they'll kill the cell. Okay, so they can make cytokines, they can kill other cells, they can steal the resources, or they can shut off the dendritic cells. Those are the four main ways that regulatory cells are going to suppress other responses. Okay, now we've covered this before, right? Where our naive cell gets activated and the sort of effector T cells, Th1, 2, or 17, are going to leave the lymph node and try and go do their job. But whatever kind of response you get, you also get follicular helper cells. And so they start off, the very earliest ones go towards the B cell zones. And this is where you have that initial B cell getting activated. They meet at the junction, have their first kiss, and that tells the B cell, go form a germinal center. Well, that also tells this pre-follicular helper cell to also go to the germinal center. Okay, so they're both going there together. From that germinal center, you get somatic hypermutation, affinity maturation, isotype switching, but you're also getting, at the end of it, memory B cells and plasma B cells. If these cells, if you didn't have these, then that B cell would go looking for it, and it would actually, this is sort of drawn incorrectly, it would keep going this way and form a short-lived plasmablast. I think many times immunologists need a lesson in graphic design. That's not where the plasmablast should be. It should be way over here. Okay, so that's why early during infection, if you don't have T cell help, then you get short-lived plasmablasts. Those make IgM, enhanced complement activation. If you do have T cell response, then you get migration, both the B cells and the follicular helper cells, to the germinal center. And so this is sort of, no matter what kind of response you get follicular helper cells, and then just so you can direct the antibody response. And those are controlled by a number of different things, but I just want you to, you don't have to know any of the little details on this slide. What I just want you to know is that there's this transition that the cells will initially get activated, And so if it is a Th1 biased, it's going to become a Th1 cell expressing Tbet. Okay, that effector cells are going to turn off the signal that tells it to become a follicular helper cell, but some of them will become follicular helper cells. So a Th1 response, you get Th1 biased T follicular helper cells and so on. Okay, so I'm going to end there, and that's because you could study the different types of helper cell biases that there are, and many people have made careers on that. I think it's for the purposes of this class, it's easier just to think about what kind of infection is it, and how does that lead to the right cellular and antibody response. And so you have, what's the initial T helper bias of the response? Well, it's what PAMP receptor is recognizing it. The dendritic cell type, which influences which kinds of co-stimulatory signals are there, and what cytokines are being made that influence the T cells. So on the test, I can guarantee you that I'm going to ask you about how does a T cell become programmed to be Th1 cell? Well, you have initially IL-12 signaling, but then interferon gamma signaling that gives you Tbet. If you have IL-4, that's going to signal to give you GATA3, give you more IL-4 to reinforce that pathway. And then I'm going to ask you, how do those different, once you get those different biases, how do they actually work? What is the main job? Is the Th1 cell going to activate T cells and macrophages, kill intracellular things? So Th2 cell, it's going to direct IgA IgE production and granulocyte activation to kill an extracellular pathogen. Okay, and that's where I'll stop and I'll take questions. Excuse me. Well, there's a question, are mutation in cytokines or cytokine pathways being used to study coronavirus? And can it be applied in this case? And the truth is absolutely yes. It's not clear why some people suffer very severe disease from coronavirus and others do not. But one hypothesis is that absolutely this could be, how much of a bias do you have towards a Th1 response, or how much of a bias are you having against a or towards a Th2 response? Obviously, having a Th2 bias makes it worse, right? Because it gives a handicap to the T cell response. So the question is, the short-term plasma cells, the primary foci, are made in the T cell zone. Yes, they're in the paracortex. Okay, if there's no other questions, I will see you on Tuesday. Tuesday will be the first day that we're trying to put it all together. When is everything happening?